Good day, learners. Welcome back to our chemistry class. Today is going to be the continuation of our qualitative analysis. If you could recall in our previous class, we have discussed on cations, that is the identification of cations, and the reagents we use to identify cations. Today, we are going to be looking at identification of anions. Before we proceed in our class, if you are a new member on this channel, kindly subscribe to my channel and turn on your notification bell for my subsequent content. For our beloved members, don't forget to share, like, love, and also leave a comment as we move on. So, let us proceed. Table for identification of anions. Before we talk about the identification of anions, there are some reagents that we normally use to identify anions, the bench reagents. One of them is silver triosonitrate 5, followed by dilute HNO3, followed by dilute HCl, followed by lead 2 chloride or lead 2 nitrate, also barium chloride. Now, these are the reagents that we use to identify anions. So, let us now move on step by step. Number one, test for chloride ion. Chloride is when chlorine gain one electron. It's from Cl raised to power minus. Now, the first one is to test for chlorine. A, sample solution plus dilute HNO3. Solution means that water has been used to dissolve the solid sample. So the resulting one is what we call solution. Now that solution, now it was taken, then dilute HNO3 was added to it. If you observe no visible reaction, So in that case, there's no inference for that because we don't know anything that is there. We can't suspect anything. Now, plus silver triosonitrate 5. When you now add silver triosonitrate 5 to it, and it gives white precipitate. That is the observation. White precipitate was formed. It means that chloride ion present in that solution. Second part of it. The precipitate that is being formed from these A parts, adding aqueous ammonia to it in excess. When you add aqueous ammonia to the precipitate that is being formed here, the precipitate dissolved. When you get it dissolved means we cannot find the droplet of particles there again. Everything dissolved. That means that chloride ion confirmed. Meaning that we are very sure that chloride ion is present there. The third one, another experiment is that precipitate that is being formed in this A exposed to sunlight. When you expose it to sunlight, the precipitate darkens, turned green. That is, it's turned to color gray. It means that chloride ion was confirmed as well. That is test for chloride ion. Second one, test for SO42 minus, tetraoxosulfate 6 ion. The first part of it, sample solution given to us plus barium chloride. When you add barium chloride to it and you have white precipitate, when we have white precipitate, the inference means that CO32 minus, SO32 minus, or sulfate ion suspected. So this SO3 is what we call sulfite ion, while this one is called sulfate ion. These three were suspected. Now, for the P part, this white precipitate that is here, you now add dilute HCl to it in excess. We now found that the precipitate was insoluble. It means it remained the way it was. It was insoluble. That means SO42 minus confirmed. That is the inference. So, though these three were suspected before when we added barium chloride, but when we now had dilute HCl to it and it remained insoluble, it means that only sulfate ion was confirmed in that solution. The third one is test for SO3 2 minus, that is sulfite ion. The first part, to your sample solution, add barium chloride to it. So when you add barium chloride to the sample solution, and the observation was white precipitates, it means CO3 2 minus, SO3 2 minus, or sulfate ion suspected. That is one of them. The second part, this precipitate that is formed here plus dilute HCl in excess. The precipitate dissolved, colorless gas with irritating smell evolved. The precipitate dissolved, colorless gas with irritating smell evolved. The inference is that the gas was sulfur 4 oxide from sulfite ion. No, when you remove these 2 minus is for the oxygen. Oxygen has a valency of 2. So when you remove 1 O, so it's going to be SO2. So sulfur 4 oxide was the gas given off, then from where? From SO3 2 minus. The third part, solution from B. This solution from B plus acidified KMNO4 in drops, acidified potassium tetraoxomanganese 7 in drops. Gas turned acidified potassium tetraoxomanganese 7 from purple to colorless, meaning that the purple color of KMNO4 will turn to colorless. When something like this happened, the inference is that SO3 2 minus confirmed. It means that solution contained SO3 2 minus. The first one is test for CO3 2 minus, triosocarbonate 4 ion. The first part of it, to the sample solution, add barium chloride. The observation, white precipitate was formed. The inference, CO3 2 minus, SO3 2 minus, or sulfate ion suspected. B, the precipitate formed in this A plus dilute HCl in excess. Precipitate dissolved. Effervescence, colorless, odorless gas evolved. When the observation was observed that way, and sometimes, how do you get the sometimes you notice the acidity of this particular gas that is being evolved. That is when you will be provided with litmus paper, blue litmus paper and red litmus paper. So, which means that anytime you want to add HCl to a solid sample or to a solution, always be with your both blue and red litmus paper. How do you do it? You wet it with your distilled water. Wet the litmus paper with your distilled water, then put it at the mouth of that particular test tube. Then, when you are adding your HCl, make sure the HCl does not touch the litmus paper. So when you add the HCl, definitely this particular solution, either it is solid or solution, the precipitate will dissolve. When precipitate was dissolving, effervescence will occur. Effervescence means giving off of gas. It means gas is coming off. For us to now know the acidity of that gas or alkalinity of that gas, it means that we have to use that litmus paper. So the litmus paper we have to turn. If the gas has been in this inference, the gas is carbon 4 oxide from CO3 2 minus. It means that the litmus paper you put at the mouth of the test tube will turn blue litmus paper to red because the gas is an acidic gas. So you should turn that blue litmus paper to what? To red. So we can also include it in our observation that precipitate dissolved, effervescence occurred, colorless and odorless gas was evolved. The gas turned blue litmus paper red. Now come to this inference. The gas is carbon 4 oxide from CO3 2 minus. So now the C part. Gas bubbled through lime water. The gas that is being evolved here, it will be bubbled through lime water, which is calcium hydroxide. When you bubble it through lime water, gas turned lime water milky. So this is one of the observations of carbon 4 oxide. So CO3 2 minus confirmed. But in our WAEC examination, this might not be given. But during our undergraduate level, we are going to do this in our higher institution. So let's proceed. Number 5, test for HCO3. Test for hydrogen triisocarbonate 4 ion. A solid sample. Now we have a solid sample this time around, not a sample solution. Solid sample plus dilute HCl, the aqueous one, is give effervescence, colorless, odorless gas evolved. And don't forget, as I said on number 4, whenever you are using HCl, make sure you go along with your litmus paper, blue and red litmus paper. So when you are using HCl, make sure you go along. Secondly is when you are eating, whenever you want to eat, anytime you want to eat, make sure you use your blue and red litmus paper. You wet it. After wetting it, you put it at the mouth of your test tube. So when you are eating, when gas is being evolved, well, that is going to tell us the nature of that gas. Now, you can do that here as well. Now, this one, effervescence, colorless, odorless gas evolved. Then CO3 2 minus, HCO3 minus suspected. The second one, the gas that was evolved here bubbled through lime water. Gas turned lime water milky. So this is a confirmatory test. CO3 2 minus, HCO3 2 minus confirmed. So from here, definitely CO2 gas must be evolved from here because carbon 4 oxide can come out from this place. Third one, sample solution plus dilute HCl. Effervescence, colorless, odorless gas evolved. CO3 2 minus, HCO3 2 minus were confirmed in that solution. This is the last aspect, number 6, test for NO3 minus. The sample solution given to us plus freshly prepared iron 2 sulfate plus concentrated H2SO4. To perform this experiment, we are going to slant our test tube. After the addition of freshly prepared iron 2 sulfate, to add your H2SO4, you slant your test tube. So when you are adding it, there's going to be a brown ring at the mouth of the test tube. That inference is that NO3 minus confirmed. That solution contains NO3 minus. So in all this that we have discussed, in our WAEC examination, mostly we will be tested with chloride ion, CO3 2 minus, SO4 2 minus, and that's the major 3 that we will be tested for. Then sometime we used to test for HCO3 minus. So in this case, this is the theory part of qualitative analysis. So in our next class, we are going to move to the lab to look at how do we perform these experiments. How do we perform the dropwise solution? How do we perform when it is in excess? Then what are the color changes so that we view it directly. So I remain Latif Ismaila Akombi, your chemistry tutor and mathematics. If you are a new member of this channel, don't forget to subscribe to my channel, click the notification bell for my subsequent content, and do not leave until you like, share, and leave a comment. Thank you very much.